We just saw how amino acid based hormones are made. And so I'm just going to go ahead and also do how steroid based hormones are made. So, steroid hormones or cholesterol based hormones. How are they made? Well, this is much simpler than the amino acid based hormones. We have our cell, and within that cell is smooth endoplasmic reticulum, smooth ER. Smooth ER winds extensively throughout the cell. It can actually make up over half of the total membrane surface of a cell. And there's a few different functions to smooth ER. Smooth ER makes lipids, cholesterol, and can do some detoxification of drugs and alcohol. Because it synthesizes cholesterol, this is where the steroid-based hormones or those cholesterol-based hormones are going to be made. And we take cholesterol and pass it through a Golgi apparatus where it gets modified, if not there within the smooth ER. Now, cholesterol or steroid-based hormones are lipid soluble, if you recall. And because they're lipid soluble, they cannot be stored within the cell. When that steroid hormone arises out of the other side, it passes right through the plasma membrane because it's lipid soluble. Now, amino acid based hormones can't do that. They're stored in vesicles, they're contained in those vesicles, and they can't go anywhere. But steroid hormones can come right out of the cell. Now, in order to make it into the bloodstream, a steroid based hormone is going to need to become soluble in that blood. And in order to do that, get my blood vessel here. In order to get into the bloodstream, it's going to need a protein carrier. And we call those binding proteins. And when a binding protein grabs this inside the blood, it's like a giant sinker that pulls this cholesterol molecule down into the water, making it soluble within the blood. Okay, this isn't quite to scale. But there's a red blood cell flowing through the blood. And here's a steroid-based hormone. Say testosterone or estrogen, cortisol. You can choose whichever one. And attached to it is going to be a binding protein. For example, testosterone binding protein. This is going to allow it to be transported to its destination. Now, because of this quality, that they're lipid soluble and they can get right out into the extracellular fluid, and if a binding protein is available, be taken up into the blood, we say that the blood value of a steroid hormone, what we measure in the blood for that steroid, does reflect the rate of synthesis by that cell. How much and how quickly the cell's making that steroid hormone. If it makes more of it, it's going to make its way into the bloodstream. Whereas amino acid-based hormones can be stored, steroid-based hormones cannot. So basically, the smoothie R makes cholesterol, and then somehow, either in the smoothie R or by the Golgi apparatus, cholesterol is chemically transformed to the steroid hormone. Pretty simple process. It does not involve the genes, at least not for the cholesterol component. Okay, because these are lipid soluble, it also means that they don't have free rein to travel throughout the body, throughout the water environment, which means that metabolizing these can take longer. Excreting them can take longer because they can pass right back through cells and get back into the body. Those renal tubules that would otherwise filter out products from the bloodstream and then dump them into the urinary bladder for excretion, these molecules can move right across the cells and back into the body. So steroid-based hormones tend to have longer half-lives than amino acid-based hormones. A half-life is the amount of time that needs to go by for 1/2 of the original product to still be present. A second half-life would be now half of that amount yet again, whereby after 2 half-lives, you only will have 1/4 of the original amount of this hormone still circulating. These are going to have longer half-lives in the body, but they cannot be stored. And so the body needs to make them when they're needed. And they'll move right into the bloodstream. And we can measure those in the bloodstream to know the rate of synthesis. Not so with amino acid-based hormones. So these are the steroid-based hormones and how they're made.